Helen Michaels, Associate Professor at Bowling Green State University. Today, I'm really excited to tell you about the master's thesis work of Meigan Day. This work builds upon a data set that was collected by my research collaborator, Dr. Ryan Walsh, who's a conservation biologist at the Toledo Zoo. This photo shows the Nature Conservancy's Kitty Todd Nature Preserve, a 1,400-acre property shown in this location by the red arrow. And this property has the greatest diversity of rare species of any protected natural area in the state of Ohio. This site is also among the best examples of an oak savanna ecosystem, which once covered much larger areas of North America, but by the mid-1980s had dwindled to less than 0.02% of its original extent due to urbanization, agricultural expansion, and fire suppression. This area and other protected oak savannas in the Midwest have been the focus of special efforts to restore habitat for pollinator conservation. And these efforts have typically involved prescribed burning activities, selective removal of woody species to maintain a semi-open canopy, and planting native savanna species, particularly to increase abundance of the butterfly host plants that have been species of conservation concern in these areas. Now, these habitats are also typically assessed for floral resources as an indicator of adult pollinator resources based on estimates of flowering stem density. A great deal of these efforts have specifically been motivated by habitat restorations needed for conservation of the Karner blue butterfly, an oak savanna indicator species for Midwestern oak savannas. Federally endangered since 1992, the Karner was once scattered throughout the Midwest, and captive breeding programs at the Toledo Zoo succeeded in reintroducing this butterfly into several sites starting in the state of Ohio in 1998. Now, this butterfly greatly prefers the semi-open canopies that promote the development of its host plant, the wild lupine. An additional factor that has found to be believed to be important in the success of the larvae of this endangered butterfly are symbiotic ant species, which are believed to protect the larvae from attack by predators. This restoration work for the Karner blue has also been complicated by the unusual life history of this species. It's a bivoltine butterfly producing 2 generations per year. In particular, Ryan's efforts were trying to identify what environmental factors might have impacted the probability of Karner blue site occupancy following a devastating 2012 drought. So these surveys, which were done throughout the spring and summer of 2016, categorize the different locations according to whether they were currently occupied, formerly occupied, that is, prior to 2012, but not afterwards, whether they had been release sites or were currently designated as priority restoration sites for future reintroduction work. And in all of these sites, a total of 15, Ryan quantified the density of ant nests, the densities of flowering lupine, and a wide variety of other environmental factors that were measured. And surprisingly, it was the density of ant nests and flowering lupine and nectar plants. They all did increase the probability of occupancy. But the surprising part was that they didn't explain as much variation as the heat loads in these sites. Now, given the influence of microclimate on the probability of occupancy results that he found, that also got Meigan and I thinking about what's the effect of environmental factors in these different types of sites on nectar quality. And furthermore, is there some component of what species are present in these sites that might be influencing the success of the Karners? Meigan approached this by building on Ryan's data set, which had characterized the numbers of stems of each species in quadrats that were in flower in these sites. Now, because she couldn't possibly characterize all species from Ryan's vegetation survey, she focused on those species that were occurring across multiple sites and at higher densities. She ended up characterizing a total of 22 flowering species, illustrated here, that were selected for specific evaluation on nectar quality and environmental factors. And because she had, as a graduate student, limited time and limited gas money, we ended up focusing in on collecting data from field sites, primarily in Ohio. So in the populations Meigan sampled, she laid out 25-meter quadrats, which was the basic sampling unit that had been in Ryan Walsh's surveys. And wherever she encountered any of her 22 target species, she counted the numbers of open flowers on a stem and collected data on soil moisture, air temperature, humidity, and canopy cover. Flowers were then bagged following collection of these environmental measurements, covered to prevent visitation by flower visitors, and then she came back in 24 hours to collect her nectar samples. Now, after retrieving the flowers, they were transferred either into microcentrifuge tubes, or in some cases, like a flower that would be relatively large, she was able to sample the nectar directly with microcapillary tubes. Whenever she had them in microcentrifuge tubes, she used this nifty field portable microcentrifuge that was powered by a power inverter and her trusty Prius in order to pull down the nectar through a glass wool filter, which nicely separated the pollen from the nectar and enabled her to extract the nectar sample below without contamination by any pollen coming through the sample. That nectar sample was then placed on a refractometer, which allowed her to determine sugar concentration in these samples. So this information from the field, the combination of what was in the nectar and the numbers of flowers per stem that was in the ground in her field sites, was used to determine a species-specific nectar and nectar volume and sugar concentration on a per flower basis. So that nectar volume and sugar concentration on a per flower basis would then be combined with her estimates of floral abundance for each of the species in the field in order to determine the contribution of nectar sugar on a per stem basis for each species. Now, that average sugar on a per stem basis was then combined with the vegetation data that Ryan Walsh had collected in order to determine on a quadrat level basis the contribution of any individual species stem in the ground, given its abundance in any of these quadrats, enabled us to determine a species-specific contribution on a per quadrat basis to the nectar resources present at a given site. We used those data on the species site contribution to combine that for each of the species that might have been present in a single quadrat to evaluate nectar resource availability within each site for each of the spring and summer seasons when Karners would have been flying. So we used this information to primarily answer initially two questions. How do environmental factors affect nectar composition? And can nectar quality be reliably associated with species identity? Now, it turned out that when she looked at her environmental data, all of that work to measure soil moisture and air temperature didn't help predict anything. And so those dropped out of our models. However, it turned out that humidity increases were associated with increased nectar volume, which would be expected because that would affect evaporation rates from the flowers. And again, as you might expect, canopy cover would have been a significant factor in predicting sugar concentrations, because more canopy cover, less light, less photosynthesis would have been a reasonable predictor to begin with. Surprisingly, the strongest predictor of what was found in any particular flower was species identity, consistent with literature suggesting a strong genetic component as well to predicting sugar in nectar. We then wanted to use our data to ask some questions that might be of value for restoration planning. So we wanted to know which species contributes the most nectar volume on a per flower basis, and therefore might prove to be important in supporting pollinators during warming and drying periods. And we found that for the spring season, dewberry was the most important source of nectar volume. And for the summer season, Baptisia tinctora was the winner in that case. And in both of these cases, these are relatively large flowers compared to the flowers at this low end of the scale, which tend to have relatively small flowers or small florets contributing to the overall floral display. We also wanted to know who's making a lot of sugar on a per flower basis. How concentrated is the sugar within each flower? And in that case, there were 4 species that all kind of jumped out as being important contributors to sugar concentration. So dotted horsemint shown here, one of the Liatris blazing star species, butterfly milkweed to be expected, real popular with pollinators, was also one of the winners here. And finally, bee balm, Monarda fistulosa, was the 4th of the 4 that were most important. Factoring in these characteristics on a per flower basis, combining that with our field estimates of floral abundance, which species is contributing the most sugar on a per stem in the ground basis? That was what we were really trying to get at, to try to extrapolate this to a site or an area on the landscape level. And that analysis turned out to be very intriguing. It found that Ceanothus americanus, New Jersey tea, was far and above the biggest sugar bomb in a quadrat. And that's not too surprising. It's somewhat to be expected. It's a woody shrub. And as you can see, there are lots of little flowers here, each of which had a decent amount of nectar. But the overall floral display, if one of these plants is present in a quadrat, had a disproportionate effect. There was much, much more sugar available in that position in a particular site. So we then wanted to move to ask, how do nectar resources vary between these sites and between the seasons? And we found a really interesting result. This figure shows sugar per quadrat. So it's like a sugar resource density for some pollinators zooming in to a location. And it shows for the spring season in blue and the summer season in green here for the 4 different site categories. The 2 that historically have had Karners, either formerly occupied or currently occupied from 2016 on when the samples were taken, release sites where Karners had been reintroduced, and sites that are priority restoration sites. The really interesting result here is that for sites that had Karners in residence continuously, they had considerably higher amounts of sugar on a per quadrat basis than the release sites did, perhaps suggesting that the reason why Karners did not stick around in those sites for very long was that they simply didn't have enough food, particularly in the spring broods, to support their needs. Similarly, for the summer, when we look at sites where Karners have been in residence for any amount of time, they didn't have a huge amount of sugar. But release sites and sites that are being targeted for restoration have lots of floral resources. So To conclude, we think that combining vegetation data with nectar sugar per stem can suggest some new insights on restoration for pollinators. And although lots more work needs to be done, this work suggests that higher spring nectar resources may be particularly important to Karner blue population persistence, and that it's essential to assess nectar resource dynamics between the seasons. And we view this really as a first step to guide species selection for restorations and assessment of future sites for restoration work. And more important than anything else, we need to develop a catalog of nectar resource estimates. And that's not just sugar, of course. We know there's amino acids and all kinds of other goodies in nectar that could be important to pollinator success of native oak savanna flowering, what's present in these native oak savanna flowering species, and the pollinators that depend on these particular habitats. So I'd be happy to answer questions in any fashion, email, or a subsequent opportunity to interact. But before I sign off, I want to give a special shout out and thanks to my colleagues, Kevin McClooney and Karen Root, whose input into the development of this work was essential, and to some of the people that helped Meigan get the data, and to some financial assistance from a Barbara Long Masters Award from Bowling Green State University.